Welcome, everyone, to MiceTech Talk with the technical information scientists of the Jackson Laboratory. My name is Janine, and I'm happy to serve the research community by answering your scientific questions.
And I am Xintong. MiceTech Talk is a way for us to connect with research community and answer some of those mouse-related questions live.
All right, so hopefully you guys are ready for MiceTech Talk because it's ready to begin. We're ready to begin. Today, let's talk scientific sessions in oncology.
Yes, we have two scientific sections on oncology research that was scheduled last Wednesday, and there's one upcoming this Wednesday. And for today's topic, we will discuss the latest developments in oncology research, and which was shared during the April 7th scientific session.
At the scientific session, we had 3 JAX staff speakers describing data from our in vivo pharmacology services team out in Sacramento, California. And Xintong and I thought it would be fun to summarize our learnings from that session. If you guys find this content intriguing, the good news is that we have linked the registration for tomorrow's April 14th session below in the video description or the comments field of the video. Also, for any of you watching live, go ahead and use the comment section of the video to tell us where you're viewing from and any questions you have today, and we'll try and answer those.
Yeah, so first question, Janine, why should we care about the age of the mice we use on our studies, specifically for the oncology research?
Yeah, Xintong, you're referring to the first of the 3 talks in which our senior study director, Aaron Rose, talked about using aged Black6 mice for syngeneic oncology studies. So for those of you who may not know what a syngeneic oncology study is, that's one in which you take a mouse tumor cell line and then inject that back into a mouse of the same genetic background. So we know generally that cancer is a disease typically for an older population. And we also know generally that immune function will change with age. So for instance, particularly for immuno-oncology research, some checkpoint inhibitors actually work better in aged populations. And this is hypothesized to be because of declines in immune function. In contrast, the majority of syngeneic studies are done in young mice about 6 to 12 weeks of age, which corresponds roughly to an 18-year-old human. And so if you wanted to start to think about expanding your applications to a translational application and correlate human age to mouse age, you might want to start modeling mice that are corresponding to a person in their 50s, 60s, or 70s. And you actually would need to use a mouse that's around 78 weeks of age or older. So our group in vivo pharmacology services demonstrated in 2 different C57 Black6 derived cancer cell lines that they did indeed see differences in anti-PD-1 responses in young versus middle or aged populations of C57 Black6J mice. They could vary the numbers of cells injected and the dosing regimens, but overall, the take-home message was that the middle and the aged populations were actually more efficacious in this type of treatment. Something else that they noticed was that there was evidence of cachexia in the older mice, whereas in the younger mice, the mice just were super healthy and continued to gain weight. So my big take home from this was, obviously there's still value in using young mice for mechanistic, proof of concept, and screening type of studies. But if you're starting to look for more translational data or starting to look at complexity and comorbidities associated with aging, you could also consider adding, an aged population to your therapeutic antibody pipeline. So I just wanted to quickly mention hi to Esther again. Good to see you again. And Adil from Turkey and John in Bar Harbor. Xintong, what were your impressions on the sessions from last week?
Yeah, a lot of exciting sections. And I mean, I would have to say I agree with you. We should be thinking about to use the aged mice for oncology research. I mean, if I were able to go back to my PhD study, I would be happy just to use more aged syngeneic models, to make my results look more translatable. So one exciting topic I have learned from this section is that we are developing the NSG SGM3 IL15 double transgenic mice. And as some of you may be aware, we do have the NSG SGM3 mice, which expresses the human interleukin 3, CSF2, and kite ligand combined with all the features of NSG mice. So the SGM3 mice actually allows better engraftment of myeloid cell lineages. And we also have NSG transgenic IL15 mice, which expresses the physiological level of human interleukin 15. So this IL15 transgenic NSG mice would allow better engraftment of nature killer cell. And here in this session, the Basile reported the NSG SGM3 IL15 double transgenic mice. This is a good model which will develop robust myeloid and NK cell lineages at the same time after the engrafted with human hematopoietic stem cells. Since we all know the myeloid cell populations, especially dendritic cells and nature killer cells, they are both key effectors of the innate immune responses, and they are critical in the activation of adaptive responses. A lot of papers or reviews actually mention that those cell types may interact with each other during the immune activation process. I do think this model would be a great platform to evaluate the interactions between the myeloid cell and NK cells. And also this model can express a relevant physiological level of T regulatory cells, and which can also be a useful model to learn the therapeutics which targeting the T regulatory cells as well. And another exciting topic I learned from this section is about the PBMC humanized mice. So both Dr. Brian Soper and Dr. Basile Siwi discussed the PBMC humanized mice for oncology study. Hey, Janine, would you comment on the PBMC humanized mice for OncoHeal study?
Yeah, sure. Thanks, Xingtong. Yeah, it's interesting to comment about your SGM3 and IL15 description of that humanized mice, because I think one of the challenges with mouse modeling is trying to develop models that better reflect a human condition. And a lot of people are interested in seeing kind of the interplay of several different immune cell types. And so our modeling techniques have to get more and more complex to adjust for that. So to get to your question about the PBMC models that were talked about last week, PBMCs refers to peripheral blood mononuclear cells, and those are cells that are taken from adult human blood donation. The PBMCs end up being a pretty convenient source of cells, human cells, to study T cells and T cell responses, and they're very functional in that capacity. So one challenge with these models is, again, the theme of trying to improve translational application in a mouse model, is that those human T cells have been educated in a human thymus. And when you're transplanting that into a mouse, even an immunodeficient mouse that can take those human cells, those T cells now are going to see the mouse as a foreign invader. And so those T cells will start to recognize them and try to eliminate them and attack them. And what happens to the mouse is for the mouse to develop a graft versus host disease response. So the graft of the T cells is now attacking those mouse tissues. One way to mitigate this is to engraft the tumor or tumor cell line into the NSG mouse first, and then to engraft the PBMCs after, so that the amount of time that the T cells, the human T cells, are in the mouse is shortened. And then the other way to do this, which was described in the sessions, is to reduce the incidence of graft versus host disease by modifying the NSG mouse hosts to knock out important molecules that are involved in the graft versus host disease response, which are MHC class 1 and mouse MHC class 2. So one study that Basile presented during his using this PBMC platform was to test the efficacy of a molecule called a bispecific T cell engager, or a BiTE. And these are really popular drugs that are being developed now. And they're generally structured so one side of the molecule has a binding site for your tumor target of interest, and then the other side of the molecule binds to the human T cells. And what this can potentially do is to bring in the T cells and the target tumor cells in closer proximity to each other, such that the T cell has a better chance to have an efficacious tumor killing or other kinds of effector responses, desirable effector responses. So in the study that Basile presented, they compared their experimental EGFR BiTE to an approved anti-EGFR antibody called cetuximab, and they did indeed demonstrate a greater efficacy using the EGFR BiTE. And this is something that they could do in this PBMC humanized model, where they could use a patient-derived xenograft or a cell line that had the human-specific EGFR target, and then they could also use the human-specific PBMCs, which are the T cells that would then be brought into the tumor. So I thought this was a really interesting application of the PBMC model to develop these novel antibodies that are becoming really popular in drug development and are actually showing a lot of efficacy. Xintong, what did you want to say about those PBMC models?
Yeah, first of all, I to agree with you. You see, the PBMC humanized mice, I mean, one limitation we learned before is because if you PBMC humanize NSG, they will develop graft versus host disease quite fast. And if you are using the double knockout, actually we can minimize the impact of the graft versus host disease, and you can engraft the tumor, and you can study your therapeutics against those tumors. I think this is really a faster way if you compared with the CD34 humanized mice platform. I also want to mention another application of this PBMC humanized model is that you can use the PBMC humanized mice, either the NSG or NSG double knockout mice, to evaluate the cytokine release syndrome, which associated with immunotherapy, such as a CAR-T or bispecific antibody, as you just mentioned about, and also the disease-induced cytokine storm as the COVID. The cytokine release syndrome, it is actually resulting from the rapid production of inflammatory cytokines. So in some severe cases, the cytokine storm can definitely result in multi-organ failure and even death to the patients. For in our in vivo systems that Brian described in this section, the PBMC humanized mice, if you treated these mice with immunotherapies, we have been tested the monoclonal antibodies as the anti-CD3, anti-CD28, or the TGN-1412, which is a CD28 superagonist. We found that the PBMC humanized mice we can detect the strong cytokine release, including the interferon gamma, interleukin 10, and interleukin 6, and other cytokines. And this is also correlated with the increased dose, and they are being released in a very short period of time. What's one advantage of this platform is that you can also engraft the tumors into the PBMC humanized mice, and then you can evaluate the cytokine storm in the most clinical relevant, translatable settings. And in this talk, again, Brian also compared the in vivo platforms versus the in vitro. What I learned from this talk is that in vivo assay can, of course, provide a systematic analysis of the consequence of the cytokine release, but the in vitro assays cannot measure. So all in all, I would highly recommend that you guys should be, if you have time, you should register and join us on this Wednesday for more detailed data of our cytokine release syndrome platforms and other models just mentioned.
Yeah, great. Yeah, Xintong, the point that you made about the in vitro versus in vivo systems, I think was a really good one for CRS. T-Genero, which the molecule you described previously, hadn't been tested in mice prior and ended up, once it went to clinic, getting this huge reaction from cytokine storm, which was really interesting to see now that we have these models that you can actually could have potentially predicted that in a mouse model. So that would have been, a potential place where in the drug development pipeline, using the in vivo model potentially could have helped identify that sooner.
Yep.
Okay, great. That's probably all the time we have for this week. Xintong, do you want to describe the resources we've linked below?
Yeah, of course. So I have provided a link, should be the first one, which is allows you to register for tomorrow's section. It's a 2-hour section, and with all the talks will be related to syngeneic mouse models, the PBMC humanized mice, and also about the NSG SGM3 IL15 double transgenic mice as well. And in addition to that, I do provide 2 links also for the NSG variants. You can find all the detailed modified NSG mice that we have from our database, as well as the cytokine release syndrome, a brief introduction to the cytokine release syndrome as well.
Great. And I think there's also, did you mention this? The Aged Mouse blog is as well. Yeah, great. Yep. All right, thanks. Well, thanks everybody for attending. Our next MiceTech talk is called Let's Talk SARS-CoV-2 Testing in Mice. And that's next Tuesday, April 27th. If you guys have been following us for a while, about a year ago, we talked about whether SARS-CoV-2 could infect wild-type mice, and the answer was no, and you needed to have a humanized ACE2 receptor mouse. Something that we've learned is it's gotten a lot more complex since then, so hopefully you guys will join us on April 27th when we update you on kind of some new findings in the literature. So if you liked today's content, please join us again on LinkedIn and YouTube. You can like and subscribe to this video and to MiceTech Talk, and you'll get notified before the session on April 27th. So this is Janine and Xingtang saying stay healthy, stay safe, and stay excited about research. All right, thanks guys.
Bye-bye.
Bye.